Welcome back again. This time we're going to talk about membrane transport in the cell. And we've mentioned the basics of this previously when we learned about the cell and anatomy. But I want to hit a few more things and cover some new material as well. When talking about passive movement across the cell, that's passive transport. And we use the word diffusion when things are just moving down their concentration gradient. If it's the type of molecule that requires a protein channel, then we call it facilitated diffusion. And this simple diffusion, the rate of the diffusion is going to depend on a bunch of different factors. And these include the temperature at which the reaction's occurring, the molecular weight of the substance that's trying to diffuse, how steep is the gradient. It's going down, but is it going down a little slope or is it going down a steep slope? How much surface area is there for things to cross? And lastly, but not least, the viscosity. It's harder for molecules to move their way through a thick extracellular matrix than a watery, thin extracellular matrix. And then let's just make sure you understand the difference between diffusion and osmosis. In diffusion, we're talking about a solute, a molecule crossing a semi-permeable membrane, and eventually at equilibrium, you would have the same concentration on both sides of that. In osmosis, we are talking about water molecules. So yes, there can be solutes on both sides of it, but those solutes are unable to cross the membrane to reach equilibrium. So what is going to move is the solvent, the water. So the water is going to move from the side of low solute concentration, where there's more water per molecule, to an area of higher solute concentration where there's less water per molecule. And that's going to happen until the concentration, as you can see in the picture on the far right, is exactly the same between the two sides of the semi-permeable membrane. But what you should notice in this one is that the volumes are not the same anymore because water has moved from the left side of the container to the right side of the container. All right, now let's talk about some active processes. And I don't want to talk about movement of big molecules that undergo phagocytosis and pinocytosis and exocytosis. I want to stick to things that are smaller molecules that are going to require active transport. So the type of active transport that you learn about in a traditional introductory biology class and in anatomy is actually primary active transport. We still have a carrier protein required, and we are moving substances instead of down a concentration gradient. Now we're going up across concentration gradient, and we're using energy directly from the breakdown of ATP. So in order for this to happen, ATP is going to break down to ADP and an inorganic phosphate. So we're going to have some phosphorylation of that carrier protein. And when that carrier protein is phosphorylated, it's going to change its shape, and that is going to allow the solute to move across the membrane. Now, what type of enzyme, category of enzyme have we been talking about that does phosphorylation? Stop and think about that for a second before we move on. There are a couple examples of primary active transport that we call pumps, okay? And basically they're pumps for ions. And there's two of them, of which one of them you should know really well from anatomy. So let's do the one you don't know first, and it's actually simpler, and that is a calcium pump. And a calcium pump just moves calcium ions against their concentration gradient. And I don't know if you're aware, but we have more calcium inside the cell than we have outside the cell normally. So we are moving calcium from inside to, we have more calcium outside the cell than we have inside the cell. So using this pump, we are going to use energy to move calcium outside the cell. And this is really important in the erythrocyte, because if there's too much calcium in the cell, then the erythrocyte becomes rigid and can't get through all your little nooks and crannies. But the one you know a lot about is the sodium potassium pump, because it's moving sodium up a concentration gradient and also moving potassium up a concentration gradient. So it's a dual pump where both things are moving uphill. And it's not an even exchange. It doesn't have anything to do with plus charges and minus charges. It's actually 3 sodium ions for every 2 potassium ions. And there's an enzyme here that has a specific name. So yes, it acts like a kinase, but it's called sodium potassium ATPase. Okay. And what it's going to do is going to split ATP to power this. And we talk about the 1-2-3 ratio in the pump, and that takes 1 ATPase, 1 sodium potassium ATPase, 2 potassium ions, and 3 sodium ions. And that's what gets exchanged. As I've mentioned before, this pump is going 24/7 to maintain this, this gradient. And in doing so, cells can have thousands of these, and they can use almost half the energy that a cell produces. So sodium potassium pump is super important, and not just the couple of places that we mentioned it in class. Let's look at the pictures to see exactly how this works. I'll walk you through it, but you don't need to know the details of me walking through it, but I want you to see how this actually works. So what happens is we have this transmembrane protein, and cytoplasmic sodium actually binds to it. Okay. And once we have 3 sodium ions bound to it, that actually stimulates the sodium potassium ATPase to split the ATP, generating an ADP and an inorganic phosphate. And that phosphate attaches to the pump itself, or phosphorylates the protein. And so once that protein is phosphorylated, it changes its conformation. And now the opening, instead of being on the inside of the cell, is on the outside of the cell. So the sodium can leave the transmembrane protein. And now, because we have a lot of potassium on the outside, it can then enter the transmembrane protein and bind to its binding sites, which are different than the sodium binding sites. And once we have 2 potassium ions bound to it, that actually causes the protein to dephosphorylate, to lose that phosphate. And so once that phosphate has fallen off, then that transmembrane protein goes back to its original shape, which had the opening inside the cell, and so potassium can fall out. And then the cycle repeats itself. All right. Now for something totally new, secondary active transport. And sometimes it's called co-transport. And what this is a simultaneous movement of 2 molecules. But remember, primary active transport with sodium, potassium, they were both going up concentration gradient. So this is different. And we have 2 types. A symport type of secondary transfer is when the 2 molecules are going the same direction, like if they are both extracellular and they're coming intracellular, or vice versa, as opposed to an antiport, which means they're going in opposite direction. Okay, so let me show you how that works. Okay, in coupled transport, co-transport, secondary active transport, all of these words mean the same thing. One substance is going down its concentration gradient. And as you recall, we talked about before, there's a lot of potential energy in that. And that energy is being used to allow the second substance to go up a concentration gradient. So whereas that sodium potassium pump had 2 ions going up a concentration gradient, and it was powered by ATP, these are powered by ATP. One is going down, and that is powering the one that's going up. And so you're going, well, what can power this? And sodium ions are the usual power. 99+% of our sodium is in the interstitial fluid. There's very little of it inside the cell. Okay. So this is a very steep gradient, which gives us a huge potential energy. And when sodium moves into the cell, it's going to give us a tremendous amount of kinetic energy to move a substance up its concentration gradient. And so one example of this is with sodium and glucose, where both sodium and glucose, where sodium has a high concentration outside the cell, and it's going to go down and enter the cell and generate a lot of kinetic energy, which allows a high concentration of glucose to also occur in the cell, because we have low glucose outside the cell. And now we're going to move it up the concentration gradient where there's already a lot of it. So this is an example of a symport where the sodium is going down the concentration gradient and the glucose is going up the concentration gradient. So this is secondary active transport or co-transport or coupled transport. Another example, but one that's not a symport, would be an example of an antiport, where things are going to move in opposite directions. And we'll learn about this. This next one happens in the kidneys and in your intestines. And that is where sodium enters the cell going down the concentration gradient, and hydrogen ions exit the cell going up their concentration gradient. So we'll come back and revisit these later in the semester. And that's it for this section. So once again, thank you so much for all your hard work, and I will see you soon. Bye-bye.